Hello everyone. Today we'll be studying staining methods. Now we know that bacteria, we need the help of microscopes to study them, and in the unstained stage, it's very difficult to see the details. So how do we see those details? We see them with the help of stain. Now everyone has done staining in your first professionals in the physiology class. So everyone knows you make a smear first, right? Okay. Then we fix them. Why don't we just make a slide and start against the staining process right away? Because when we wash it, the smear will also be washed off in water. So when we do staining, first we have to fix that smear onto that slide. Now there are two methods of fixation. First is called the heat fixation. Heat fixation. Now, what is the purpose of fixation first? Let's see. Fixation 1, the slide firmly adheres, the sphere firmly adheres to that slide. Second, it becomes more recipient to staining. Third, it resists washing away. Okay. So, how much should you heat? Let's see in this diagram. So if you're making it from a liquid media or a liquid specimen, you just take a drop or so, put it on the slide. Okay, put it on a slide and let it air dry. And then you pass it through the flame twice or thrice, or till the point it is sufficient to be borne at the back of your hand. Okay. Second, अगर solid colony culture से हम बना रहे हैं, तो पहले एक drop water or normal saline is put, then we transfer a colony over it, make a smear and let it dry. पहले air dry, जब सूख गया, then we pass it through the flame a few times and till the back of our hand can bear it. So that's the end point. इतना गर्म होना चाहिए बस, ज़्यादा गर्म नहीं करना है. That is your heat fixation. Next is your chemical fixation. Now, when you prepared blood smears to do your DLC, so did you pass it through the flame? No. What did we do? We do a chemical fixation. Okay, one of the reagents which is used for chemical fixation is your methanol. So now coming on to the staining, there are many types of staining methods. First, we'll start with the simple one. That's called a simple staining. Now in simple staining, let's see. Okay, once more only, I won't repeat it again. You have a medium or a specimen, you spread it on the smear slide, make a smear out of it. You let it dry in the air first, then you pass it through the flame a few times. Now your slide is ready to be stained. Now there are two stains which can be used as simple stain. Why they called simple? Because all organisms under them would take up the same color. So you have a methylene blue and a basic fuchsin. So blue, it would stain blue. Basic fuchsin is a pink color stain. Okay. Then you put that stain over the slide, wait for a minute, then wash it in water. Now your slide is ready to be seen under the microscope. For bacteria, I told you which objective lens we'll be using. We'll be using 100x. And it's called as oil emulsion. We put a drop of oil and see it under the microscope. So what will you see? You will see blue for blue and pink for basic fuchsin. So what is the advantage of simple stain? We can just see the morphological features. You can see their shape, you can see the arrangement. We'll be covering up all this in future classes. So for the time being, you know that simple stain means a stain, a chemical or a color which will stain everything of the same color. Okay. Now normally the stains which are used in microbiology are basic stains because the protoplasm of the bacteria is acidic. So we use basic stains. Second is a negative stain. Here negative means then it's the stain which are used is India ink and nigrosin. They are negatively charged. Even the bacteria and the organisms are negatively charged. So what will happen when they are negatively charged? It will repel. Like charges, they attract, but same charges, they will repel. So you have put the suspension stain, a negatively charged stain, and the bacteria is also negatively charged. So will it take up the stain? No. So it will appear colorless, and you will get a dark background. Okay. In simple staining, in normal staining, what we do, we use a basic stain because this protoplasm is acidic. So acid and basic, they will attract, so the protoplasm gets stained. So we see the bacteria. But in negative staining, the bacteria is negatively charged and the stain is also negatively charged. So it doesn't take up the stain and it appears as colorless. So this was helpful to see this capsule. This is a slide of CSF. And what is shown here is your Cryptococcus. This is a bacteria which causes cryptococcal meningitis. And you see here, this is a yeast and this is a budding yeast cells. And this transparent clear zone is your capsule. Okay. So this method of staining is called your negative staining. Next is your impregnation methods. Now there are certain bacteria which are too thin. You cannot appreciate them by simple stain. I gave you an example in your previous video. A spiral bacteria which was seen by dark ground microscope. So spirochetes, the causative agent of syphilis, that is stained with this help of impregnation. Impregnation means we deposit silver stain onto this organism and which helps us to visualize it. So you see all these organisms which are in the circle, they are spiral, finely spiral bacteria or the spirochetes, here thin-like. This is the causative agent of syphilis, which we call it as Treponema pallidum. This is stained by impregnation method, which we call it as Liverdi stain, and then that is for your tissue samples, and the other one is your Fontana stain, that is for fluid samples. So these are the two methods of silver staining. Okay, what is the reagent that is used? Silver. The method is called impregnation. Here fine structures, they are made thicker by depositing silver over them. And the last method of your staining is your fluorescent staining. We read it in fluorescent microscopes. The fluorescent dyes are special chemicals which absorb light, UV light. They become excited, and when they revert to they are unstable in the excited state, so they revert to their ground state, and in the process they emit light of the visible range. We read two fluorescent dyes, acridine orange and auramine. So here, this is they stain acridine, it binds to the nucleus. So in we are seeing here the slides of parasite Plasmodium. So these are your RBCs. RBCs, do they have a nucleus? No, they don't have a nucleus, so there is nothing here. Okay. So this is your malarial parasite. It has DNA, so the acridine orange has bound to the nucleus, and you can see the malarial parasite shining. So this is your fluorescent staining. So that was your simple stain and your negative staining, impregnation method, and your fluorescent stain. Now we come on to the very important part, your differential stain. Now from the name itself, differential, it differentiates. Okay. So these stains, we'll be reading three of them. The first and the foremost and the most important is your gram stain. You will be doing this in your practicals and you will be doing them for your exams. And this forms the big six and the most important test routinely done in microbiology laboratories. Gram staining. So what is gram staining? It was devised by Christian Gram. That's why the name gram, it's not for the weight. Okay. Many times when I asked in the Viva, what is gram positive mean? It weighs more than a gram. That's not the answer. Okay. And so gram staining, let's see the process and we'll have to go through it 2 or 3 times. So when we try to understand the principle, first let's have a look at the process. Now, first step we told you, we made a smear. Now, after making a smear, we fix it. Now, the first reagent that is put is your crystal violet, gentian violet, or any basic para-rosalind dye. Why a basic dye? Because the acid is, protoplasm is acidic. Okay. So first stain is your crystal violet. Now all the bacteria will take up this stain. Whether it is gram-positive or negative, they will take up this stain. Okay. Now second step, we add a iodine. Now iodine acts as the word used is mordant. Mordant means it combines with the crystal violet and make a diiodine complex. Okay. Now this will also happen in all the bacteria, whether it is positive or negative, they get the same reaction. Okay. Now comes the differentiating point, the decolorizer. Now here the decolorizer can be your acetone alcohol, okay, or acetone alcohol combination. So by decolorizer, two things can happen. Either it can get decolorized or it cannot get decolorized, right? Those who got decolorized, now how do we see them? We put a counter stain, that is your saffronin, which is pink in color. So which did not get decolorized, they appear as purple, and which got decolorized and we had to put a second stain for them, they appear as pink. I hope the process is clear. When we go through the theory again, we'll come to it in a detail now. Now, what is the theory postulated? Why certain bacteria, they retain the dye and they become gram-positive, and by the other bacteria, they lose the dye and they become gram-negative? The first is your pH theory to explain it. The pH, what did I tell you for the pH? The bacteria have an acidic protoplasm. So they say that for gram-positive, the bacteria have a more acidic protoplasm, so they retain the dye. That is one theory. Second theory, and the most widely accepted theory, is the theory of the cell wall. Now, we'll be reading cell wall in detail when we read the structure of the bacteria. But for today, we'll read one word. That cell wall has one compound that is called your peptidoglycan. Okay, so the gram-positive have a thicker layer of peptidoglycan. That's why the diiodine complex is retained here. It is not washed off by the acetone. Whereas in the gram-negative, this peptidoglycan layer is very thin. So the decolorizer helps to wash out this diiodine complex. Third is your lipid content, lipid A. This is present in the gram-negative. They have a thin peptidoglycan layer, but they have a lipid A. Now, by acetone and alcohol, this lipid A is dissolved, so there are large pores present, through which the diiodine complex moves out. And lastly, we have the magnesium ribonuclease theory enzyme. This enzyme is present in the gram-positive, so they help to retain the diiodine complex. Okay. So let's combine all of them together and see. We use the basic para-roselin dye because they have an acidic protoplasm. So in the gram-positive, when we add crystal violet, the iodine acts as a mordant. It forms a diiodine complex. Everyone clear till here? It's whether gram-positive or gram-negative, everyone will have the same step till here. Now, the differentiating point comes here, the decolorization. Now, because the gram-positive have a thicker peptidoglycan layer, the diiodine complex is retained by the gram-positive and they appear purple. Whereas in the gram-negative, because of the thinner peptidoglycan layer and lipid A, the diiodine complex moves out and it becomes colorless. So, to visualize these colorless bacteria, we add counter stain, which is your saffronin, which appears pink in color. Okay, so that is how your gram-positive look like and your gram-negative look like. This is your gram-positive bacteria and this is your gram-negative bacteria, pink in color. You have a modification that instead of saffronin, you can reuse neutral red as a counter stain for gonococci. That is one of the modifications. There are many modifications like you may not know. Okay, so let's move on to the next stain, that is your acid-fast staining. Now in history part, when we were reading, I told you about a scientist, which is your Paul Ehrlich. Okay. He was the first scientist to describe the acid fastness of Mycobacterium tuberculosis. Now, what do you mean by acid fast? Now, these are bacteria, which is your Mycobacterium tuberculosis. They are very difficult to stain. Why? Because of a compound present in the cell wall that is called your mycolic acid. Now, the mycolic acid doesn't allow the stains to enter. So when you read the theory, they say it's gram positive, but when you do a gram staining, they hardly take up the stain because of this thick waxy layer mycolic acid. So, but once it gets stained by carbon fixin, what we'll be reading by, we may heat it so that it penetrates. Now, once it is stained, it doesn't lose it, even if you decolorize it with an acid. So that is called acid fastness. Okay. So, and let's see the process here. This was first described by Paul Ehrlich, but later modified by Ziehl-Neelsen. So commonly it is known as your Ziehl-Neelsen staining or ZN staining for short. So what happens before staining? The bacteria, whether acid fast or not, they are colorless. Now you put carbon fixin. So I told you we heat it. So that is your heat method of staining. So they become pink in color. Now you treat it with an acid. Which is the acid we use? Sulfuric acid. Though originally it was picric acid which was described, but we here use sulfuric acid. The sulfuric acid, the acid fast bacteria, they retain the dye. Whereas the acid fast negative, they are decolorized. They lose the dye. So this is called as acid fastness. Okay. So, to see these colorless bacteria, we use a counter stain, that is your methylene glow. So, the original method which is described is your heat method, and that is called your ZN staining. And these are, there are many modifications to this ZN staining. The first modification here is your phenol. Mixing it with phenol, that is your cold method of staining, and this is also known as your Quinon's methods of modification. Originally described as 25% of sulfuric acid, which is used in your National Tuberculosis Eradication Program for M. tuberculosis. But we have various modifications, like 5% sulfuric acid is used for your N. leprae. Okay. 1% is used for your Nocardia and Legionella. 0.5% that is used for your parasites. Parasites, oocysts of Cryptosporidium, Cystoisospora, Cyclospora. You have eggs of Tinea saginata, eggs of Schistosoma, Mansoni, Kinongi. Okay, you have 0.5% also for sperm head and for spores. So these are the various modifications which are of ZN staining. One more modification you have, that is instead of methylene blue, green color dye, that is your malachite green is also can be used as a background counter staining. So that's a very important exercise in your practicals, and you are asked these questions, what is the concentration? So you have to remember 25% sulfuric acid is used under National Tuberculosis Eradication Program. Otherwise, another modification is also we can use acid alcohol. Here hydrochloric acid is used with alcohol as a decolorizer. That's another modification. So coming on to the last, differential stain, that is your Albert stain. So Albert stain is also to see for the bacteria, Corynebacterium diphtheria. Okay, what are we seeing in Albert stain? We are seeing these metachromatic granules. They are known by various names. It's also known as the Volatin granules. It's also known as the Babe's Earnest granules. It's also known as polar bodies. There are various names to it. Okay. So let's first understand what do we mean by metachromatic. Now, in simple stain, what did we say? We use a blue colored dye and we see blue colored organism. We use pink colored dye, we see pink colored organism, right? But metachromasia, this says we are putting a dye of one color, but because of the biological properties, we see actual visual effect is of another color. Here what we are doing, we are using a blue color dye, methylene blue, and we see bluish black or purple granules. Okay, so this phenomena is called as metachromasia, and these granules are known as metachromatic granules. Okay, so what are the stains that can be used to see them? One I told you Albert stain, the other I told you it is a blue dye, Loveless methylene blue. We can also have the Ponder stain. We also have a Neser stain. So these are the various stains which are used to see this metachromatic granules, typically described in Corynine bacteria and diphtheria. Besides this, we also have it in Corynine bacteria xerosis. We also have it in fungus, that is your Saccharomyces. We also have it for other bacteria like your Botrytis pertussis and Spirillum. Okay, so these are the three examples of your differential stains. Gram staining for your gram-positive and gram-negative bacteria, acid-fast staining for Mycobacterium tuberculosis, Albert staining for Corynine bacteria diphtheria. So after this, let's have a quick look at few multiple choice questions. So now the difference between the gram-positive and gram-negative bacteria is shown to reside in the... Now, we read the theory. What were the four components of the theory of gram staining? First, we said it's a basic dye. So, it's about the pH. Gram-positive have more acidic pH and gram-negative have a lesser acidic pH in the cytoplasm. Second, we read there is difference in the cell wall. What component of the cell wall we read? The gram-positive have thicker peptidoglycan layer, whereas the gram-negative have a thinner peptidoglycan layer. Next difference we read was in the lipid content. The gram-positive don't have lipid in the cell wall, whereas the gram-negative have lipid content in the cell wall, which is dissolved by the alcohol, by the decolorizer. Okay, there I've missed out. Let's cover it up here. That the decolorization is the most important step in your gram staining. Why? If you're asked what is the most important step in the gram staining, it is your decolorizer. If you do a little less, then even the gram negative can appear as gram positive. And if you overdo, the gram positive will appear as gram negative. So you have to be very careful in using the decolorizer. Okay. So the lipid content, because of the decolorizer, the lipid is washed out and the diiodine complex moved out. And lastly, we read the magnesium ribonuclease theory. This enzyme is present in the gram positive, which helps to bind the diiodine complex. So what is, let's have a look at the options. First, is there a difference in the nucleus? No. Is there a difference in the chromosome? No. Is there a difference in the cell membrane? No, it's the difference in the cell wall, okay, and specifically the peptidoglycan. So coming on to the next question, metachromatic granules can be stained with. We read now, what were the different stains we have read? We have read simple staining. Do we do simple staining for this? No. We read negative staining. No, that was for the capsule. We read impregnation, that was for the spirochetes. We read fluorescence. No, metachromatic granules were not for the fluorescence. What we read? Then we read differential stains. We read gram stain. That was for gram positive and gram negative. We read Zn. That was for mycobacteria. Then we read Albert stain. That was for metachromatic granules. Okay. So there were many options to Albert stain also, I told you. Do we use safranin? No. Do we use methylene blue? Yes. I told you metachromasia, we use a blue dye and we get purple granules. Crystal violet? No, that is used in gram stain. Picric acid is not used. So metachromatic granules can be stained with methylene blue. So coming on to the next question. India ink preparation is used for the demonstration of flagella, capsule. Yes, the answer was capsule. Spirochetes? No. That was Fimbria? No. Okay, India ink, that was, what is India is an example of negative staining. So, it is used for the demonstration of the capsule. The fourth question. Silver impregnation technique is used in the diagnosis of, we read, there were Livardetti stain and Fontana stain for syphilis. The Treponema pallidum, the spirochete. The answer is syphilis. No, but you should have a look at all the choices, though your answer is correct in here. But you should always have a look at all the choices. Lyme disease, again, it's caused by a spiral organism, spirochete. This is also all of the above. Now, here the answer is all of the above. Silver impregnation technique is used to stain all these spiral organism. Even H. pylori organism is also diagnosed by silver impregnation staining. So last question. H2SO4 is your sulfuric acid. Concentration used in Zn staining for M. leprae. Now we discussed the original concentration in Zn staining for sulfuric acid is 25%, for which National Tuberculosis Eradication Program for MTB. That is your concentration. But in the modifications we discussed, for M. leprae, what is the concentration? No, it was not 1%. 1% is for your nocardia and legionella. 5% is your for M. leprae. 10% is not used, 25% is used for MTB.